Our next speaker is Dr. Xihong Lin, who many of you know, who is professor of biostatistics and professor of statistics and chair of statistics at Harvard. Before going to Harvard in 2005, Xihong was professor of biostatistics here at Michigan, and she has worked substantially in the area of statistical genetics, among other biostatistics topics. And she is talking about accelerating trustworthy discovery by integrating generative AI, statistics, and genomic health. Xihong, thanks so much for speaking.
Thanks a lot, Mike and Bessie, for organizing this event. And this is an event I can never miss because Mike is so important to me. Okay, so let me start from the story. And so Mike and I, we were colleagues for 11 years. And so a couple years ago, the UW Stat Department, Biostat Department celebrated 70th birthday. When I came over, I saw this very, very nice poster on the wall of the Biostat Department that lists all the faculty members. And between 1960, when the department was launched, until 2016, Mike was number 26. And so he joined the department in 1984. And Rod was the chair. And when I joined, Rod was number 39. He joined the department as the chair in 1993. And when he was the chair, he recruited 2 people. And one is Ken, Mike's advisor, and the other is me. And we came in the same year in 94. And so this is the best decision I have ever made. 2 people, really important to me, and there's Mike Square. And so Mike and I, we co-advised this Mike. And so when he was a PhD student, and so when I was in Michigan, I did not work on statistical genetics. I thought I know almost zero about genetics. And then, so Mike, because I work on mixed models, and one day Mike approached me saying, I need some help with a mixed model. And then Mike and another Mike and I, we co-supervised this Mike, young Mike. And then Mike published 2 papers. He always said, oh yeah, I think I own him, bring me to statistical genetics. I think that is true. And I also own Mike as well. And so he had 2 papers published. And then, so this picture was taken a couple years ago at the 70th anniversary. And this is a really wonderful picture of the celebration of the department. You can see everybody wore masks at that time. And then another thing I want to highlight is at that event, and the department launched the Mike Boehnke's Distinguished Visiting Scholar Award. The first awardee was Ken. And so you can see from this picture, and so I had Ken, Mike, and Rod. And so Bamar prepared these slides. I just tried to zoom in for that slide. I thought this summarized Mike really well. And so this is Tamar's wording, Bamar's wording. So a fantastic human geneticist, an outstanding human being, an amazing colleague, educator, and mentor. I think and also a great partner, parents and grandparents, all are very true. And for example, I think Mike is the only person I know who has served on the chair of the admission committee for 40 or something years, the longest person I've ever served on an admission committee. He has helped recruiting so many wonderful students to the Biostat department here. And okay, after the event, and then Bamar invited both Mike and Ken and Bessie to her house and to celebrate. It's a really wonderful event. So Bamar gave me this picture yesterday. And okay, so now I talk about statistical genetics. So first, I think I will talk about whole genome sequencing analysis, and because Mike played an important role, and also that is important collaboration I had with Mike. And so basically, to do the whole genome sequencing analysis, we need to do a little homework, including the variance functional annotation, and make the whole analysis pipeline automated, and also deploy it in the cloud, make it affordable. So, okay, this picture was made by ChatGPT, and I said, look really good. And so this, I just put my picture, put Mike's picture, said make a picture, and two of us in Mike's kitchen. Look real, right? I think ChatGPT make a picture better than germline. And so this was in Mike's house. That was about 2010 when I came to visit Michigan. And so I stayed with Mike and Bessie in his house. And the next day, the morning, and two of us were sat in the kitchen discussing this paper and the Skype paper. And so this paper, at that time, there was no whole genome sequencing study and wasn't candidate gene study. And so this paper was later published in the following year, 2011. And so this SCAD method has been widely used. And in analyzing the real variance whole genome sequencing study, you can see the citation at that time. And then at that time, I remember, Mike, you told me in your kitchen, saying this paper, this work will be highly cited. And I did not know how. He had really good prediction. I did not have that sense, but he has really good prediction. I appreciate that. So, okay, so basically they start the whole chapter of whole genome sequencing analysis, and I work on in the last 10-something years. Okay, and so you can see there's a bunch of paper of that paper and bunch of paper published related to analysis of whole genome sequencing, real variance analysis by my student postdocs, and including we call the STAR method that incorporating variance functional annotation in the real variance analysis, but SCAD is the foundation for those type of models. And so then, so in order to do this analysis, we have to do a little homework, because unlike the common variance analysis, we have to functionally annotate the genome. So we built this favor functional annotation database that includes all 9 billion variants, basically 3 billion positions and 9 billion variants. And then, but not everybody familiar with computational biology. And so we built this favor GPT that is interface, large language model interface with the favor database. And so that create many different functional annotation, from epigenetic annotation, conservation, protein functions. And so for example, if you ask how many pathogenic variants does BRCA1 have, and then you can see that basically just like what you use for ChatGPT. If you use ChatGPT, it will tell you it's over 1,000. It make up answer. And so ChatGPT is good for general things, but not for specialized things. You need high quality database. And because Fever uses the ClinVar database, you can see it has 92. That is the right answer. And also, we also include the knowledge graph, which is quite excited about this. So basically, you can enter any gene, and then that will show all the diseases related to that LDLR gene, and then for any particular disease, that show all the drugs and targeted with that gene. So I think this will be useful for people. And so this basically indicates the gene and disease and the drug. So also, as I mentioned, that we want to make the analysis of fully scalable and also make it affordable. As you know, for UK Biobank data and all-fast data, the data cannot be downloaded. And so therefore, we want to, all the data on the cloud, we want to make it affordable. So for example, if one use a favor to functionally annotate the UK Biobank whole genome sequencing data. It has over 1 billion variants, only costs $6 to analyze the whole genome, and that costs $10. So this is quite affordable and for the community. And also, we are working on developing the whole genome sequencing analysis AI agent. Hopefully, we'll make that available this summer, and that will make things fully automated, from the developing the analysis plan to the action execution and prepare the result and then evaluate result and then human in the loop and finally generalized analysis result. So I think this is something we're quite excited about as well. And also we analyzed over 1,300 diseases and traits, and in the UK Biobank sample, including both the quantitative trait and disease trait. And so those are the major results. And so you can see for the coding regions and basically the coding variants, and they are most associated with cancer. That makes a lot of sense. And especially loss functional variants or the sense variants. And also, this is also the question we ask ourselves is, which gene are top genes associated with most diseases and traits, and look like those are the lucky ones. So for example, ASL1, and is associated with most different diseases and traits. So that is the first part. So you can see that Mike really play an important role and in the relevance analysis, especially in the early day, we really enjoy the collaboration together. So the second part is how can we integrate the statistic with generative AI and with the synthetic data. This is something I'm quite passionate about right now. So one thing, So there are two things I'm quite passionate about right now. So basically, generative AI is being transformative, not only in ChatGPT, but also transforming the research as well. So two things I'm passionate. One is the synthetic data I'm going to talk about. The other is agentic AI. So I just mentioned that very briefly, especially you can build using the long chain or long graph or the cloud skill or cloud code. And so synthetic data, in the next couple years, over 50% of data are likely to be synthetic data predicted by multiple outlets. And so many of us have seen those synthetic data. For example, the images, like the one I generated with Mike in his kitchen. And those is basically the synthetic images. And then the question is, those are not real data. So how can we accelerate the trustworthy scientific discovery by integrating generative AI and also statistics? So first of all, because the data are not real data, those are generated data. For example, if you look at the picture in the bottom here, this is observed data, and this is a synthetic data. You can see generated very similarly to the observed data, but those are not real data. So how can we leverage those synthetic data and to ensure the downstream analysis is valid, even though the generative model can be completely wrong? And but we still can ensure that downstream analysis is valid, but if the generative model is useful, and then downstream analysis will be more powerful. So this is the editor invited review article, and so which was led by my former student, my current student Armand, and also the postdoc Ruoyu. First of all, how can we generate the synthetic data? There are multiple ways. One is to use the variation autoencoder. So basically, you start from the original image, and then you do the dimension reduction and generate the latent space. That is through the representation learning. And then afterwards, you regenerate the data that use the decoder. So from encoder to decoder, that is one way. And another way is many of you are familiar with large language model. The backbone of large language model is the transformer. And so, for example, that I heard about alpha genome, and so basically use the DNA sequence to predict functionality of, for example, you can predict the cage site and the epigenetic regulatory site and so on. And so this is the other way to generate the synthetic data is the diffusion model. And for the transformer, basically the data need to be ordered. But like images, the data are not ordered. So the diffusion model I'm quite passionate about as well. So for example, you start from the original image, you gradually add noise until it becomes completely white noise. There's no training here. And so the art is a reverse process. Basically, you denoise the data until you recover the original image. So that basically is a generated model, use the deep learning model. Another way is basically a combination of different ones. So for example, you can have diffusion transformer models that generally work better. So Armand is very talented. And so one thing we started with, we generate the synthetic genome. So you can see on the left, and so this is original LD map, the LD map of original data. On the right, you can see this is the generated synthetic genome, LD, look very similar. And so he built using the transformer flow matching method. Because across the genome, for common variants, there are hundreds of millions of genetic variants. You can see work really well. So that basically can make it very, very scalable, and much more scalable than HepGen, especially work very well for underrepresented minority population. That is critically important because minority population generates a sample size much smaller, but we can generate an infinite number of genome. And also we can generate a synthetic proteinome. So for example, for UK Biobank, there are thousands of protein. You can see on the left, this is observed heat map of the UK Biobank protein on the right. And so this is generated a synthetic protein, looks really similar. So that's very good. However, those synthetic genome and synthetic proteinome, they are not real data. They are generated data. And so therefore, a question for us is how can the generated synthetic data from the generated AI model can be used for downstream analysis, but the analysis need to be robust. If the generated models are wrong, and I still can have valid inference, like a p-value, but if generated models are useful, and then have more power. So that's basically the question we try to answer. So the goal is to make a valid and powerful downstream statistical analysis, especially when the generative synthetic data models are wrong. So for example, suppose you have observed data X and Y. So so X is the genotype, Y is the phenotype. But generally, the phenotype can be limited, or genotype can be limited. So the sample size power based on observed data may not be high enough. One way is you generate the synthetic genome and synthetic phenome, and then you can basically infinite sample size, right? Sounds really wonderful, right? However, if the generated model are wrong, you can see the result will be wrong. So another approach is we call the synthetic data assistant method. So this all in the review article. So basically you can see, like for example, for UK Biobank, about 50,000 people have the proteomics data, but we 500,000 people all have the whole genome sequencing data or GWAS data. So what we do is we predict the proteomics data. Even for those people who have observed the protein, we still predict it. And why that is important? Because that can help me to calibrate. And if the generative model are wrong, well, I do the right analysis, and that can account for the fact that generative model is wrong. And so another approach we call the synthetic data augmented approach. So therefore, suppose you only have 500,000 people here from UK Biobank, 500,000 people here, the genotype from UK Biobank, but I can generate infinite number of genome and the phenome, significantly increase the sample size, right? So that is, but you have to use a careful method when we do the analysis. So now I'm just focused on this one. So basically, like for the biobank data is basically the whole genome sequencing data with the EIChart data. So one can look at, traditionally you look at one phenotype at a time in traditional GWAS study, but now with the EIChart data, and you sequence 500,000 people from UK Biobank, but you can look at thousands of disease traits and simultaneously. For UK Biobank, 500,000 people, million veteran program, 1 million, and ALPHA's 1 million. So you can see those are really large biobanks. And so the goal is to scan the genome and identify the genetic variants associated with the human diseases and traits. So why the synthetic data are useful? So for example, if you look at the body composition, in UK Biobank, that basically only observed for 30,000 people, even we have 500,000 people. And so then if we use restrict analysis using 30,000 people, not much power, right? Another example is proteomics data. Totally, there are over 100,000 proteins. And proteins are critically important because they are the drug target. And then many of them, and also protein is closest to the disease. And so in UK Biobank, thousands of proteins were measured. but they were not measured for everybody. They are too expensive. Another one is the brain imaging. So in UK Biobank, about 50,000 people with the brain imaging data. So you can see that this, especially this useful to understand how the brain works and also studies the psychiatric and brain disease diagnosis. And the major challenge here, those data are very expensive to collect. We won't be able to collect for everybody. So this is where the synthetic data come in. So this paper was led by my former student, Zach McCall, and was published in Nature Genetics about 2 years ago. So basically, if you look at the UK Biobank data like body composition or proteomics or imaging, they were missing for over 90 or 95% of observation. However, I have a lot of information I can use to predict those phenotypes. For example, I have demographic information on biomarkers and ICD-10, ICD-9 code for all 500,000 people. And so then what the goal is, we want to study the association and between the body composition with the protein and the SNP effect. And so then, but the protein and the body composition or image phenotype, they are only available for 5% of UK Biobank sample. So how can we do this analysis? And so this is where the synthetic phenotype idea comes in. So you basically use all those surrogates to build a prediction model to predict the body composition or predict the proteomics or predict the images. And, but the prediction can be wrong. But here you can see we predict for everybody, including the labeled sample, those people with observed proteomics data. So how can we build the prediction model? Multiple ways. One is you use the external weight. For example, like llama or a bird, they have external pre-trained weight, and you fine-tune them, and we'll use the distillation, and then to fine-tune it to make it work for the UK Biobank sample. The other way to do it, you split the data and use a part of the data for training. And so, for example, we use the 5,000 people to train the prediction model, and then build this prediction model. You can use any kind of model you like. You can use a random forest, diffusion model, or transformer, whatever generative model you like to use. And then you apply those weights to the whole UK Biobank sample. So basically, that is what shows here. So you can see for proteomics, only 50,000 people have available proteomics data, but I use this as I predict proteomics for everybody. And so that helps. And then the goal is I want to make a downstream analysis valid. Even my generative model is wrong. And I don't know the theory of the large language transformer. I don't know the theory of diffusion model. Perfectly, it's okay. I don't mind. Even it's completely garbage, doesn't hurt me. I always have the positive learning. So how can we do that? And so the first, the first way is just use a standard approach. For example, I have 50,000 people with the proteomics data. I just run the GWAS. That doesn't have lots of power. The second approach is to do the multiple or single imputation. So basically, for those 95% of people without body composition, I predict the y hat, and then I stagger them. And a major assumption here is the imputation model need to be correct. Okay? And so the third way is the synthetic approach, that if the generative model is wrong, I'm still okay. And so because I predict those Y, even for the sample, I have observed the Y. So I use those to calibrate, to allow me to do the analysis. I jointly analyze Y and Y hat, and even the generative model is wrong, but my analysis still be valid. So basically, how can you do that? So basically, you can see we do a joint model, Y and Y hat, and so you can see the regression coefficient, that is a key. The regression coefficient for the Y model, regression coefficient for the Y hat model, we allow them to be different. That is a key. Okay? And if the generative model is useful, then these off-diagonal elements will be close to 1. That basically means the prediction really good. And so that's why it helps. Okay? So the generative model can be completely garbage, doesn't hurt me. So basically, that is a flow. So you use a 5,000 sample to do the training, and then the leftover to do the analysis. And for the training sample, you use random forest, whatever diffusion model to predict the y hat. Then you transport the weight to the whole UK Boehnke sample, and then you do the joint analysis, generate the Manhattan plot. And so to see the result, so for example, if the generative models are misspecified, if they're correctly specified, you can see all the method work. And using the original data, standard method, what imputation was a synthetic approach. But when the model, generative model, is misspecified, and you can see those multiple imputation will fail, and however, synthetic approach still work. And so the key feature here is through this synthetic data-assisted method, we still can have valid and powerful downstream statistical analysis. Even the generative model is wrong, and we don't know the theory. It's perfectly okay. And so basically, this is, it turns out that joint modeling can be written as the AIPW estimator through an augmented term. And so this work also included the existing method, like a PPI, as a special case. For example, and sorry, I don't know why the formula disappeared. Something is wrong, my apology. But anyway, so the PPI, basically the augmented term is not right. And so it also assumes missing completely at random, and it requires the prediction model to be highly accurate. However, for synthetic approach, it requires a weaker assumption, requires missing at random, and also reach a semi-parametric efficiency bound because the augmented term is correct. And also, we extend this to the mixed model because for UK Boehnke sample, a third of the samples are related samples. So the traditional approach in this nature genetics method cannot be applied to all the UK Boehnke sample, only applied to independent sample. So how you use my postdoc, she developed this SynPalm approach, basically synthetic phenotype assisted linear mixed model applied to UK Boehnke proteomics data. And so you can see this, this is the protein, and the red is the number of GWAS discoveries, and using observed 50,000 protein, 50,000 labeled sample, and the blue is the number of GWAS discovery, and using this synthetic SynPalm approach, you can see we have more discovery. To summarize, and so this is my colleague, Sarah Fortune. She is a chair of infectious disease department. And so she wrote mixed AI at the bottom, statistics as a power. I thought this is a really wonderful way to represent the marriage of AI and statistics. So we all need to be open-minded, think out the box when embracing AI, and also empower valid downstream statistical analysis and scientific discovery using synthetic data, allowing the generative model and black box generative model to be wrong and misspecified. And so if we do statistically right, so that is that's what my view is. This is a sweet spot for statistician. That if we marry statistics and AI right, and then we can empower AI and empower scientific trustworthy discovery. And also build a scalable, interpretable, human-in-the-loop agentic AI ecosystem for end-to-end analysis, and field statistics, AI, and domain science to accelerate trustworthy discovery. Finally, thank you, Mike, and for all you have done, and for us, for the department and the university, and also the field of statistics and human genetics.